Hello. Thanks for getting here on time. We've got to wait 2 more minutes for everyone else. But in the meantime, I'd just like to thank everyone who emailed me to say that I left the "and rock and roll" off the end of my title. That has been noted, and next time will be added. Talk soon.
So Alistair, with all that rain we have now, will we see a hell of a lot of mushrooms growing up everywhere?
I would imagine so. I think we're going to be in one of the best seasons ever for collecting mushrooms. Shout out to the Queensland Mycological Society. Only costs $25 for a year membership, and you can be foraging with the best that Queensland has to offer, learning out there in the field with a big smile on your dial, covered in leeches. That's good living on a weekend. I'll talk to you soon.
All right. Well, good afternoon. My name is Professor André Drent. I'm a plant pathologist at QAFI at the Centre for Horticultural Science. That is the area also where Alistair is employed in. So today we're going to have a seminar about mushrooms. And first, acknowledgement of country. So the University of Queensland acknowledges the traditional owners in their custodianship of the lands on which we meet. We pay our respect to their ancestors, their descendants who continue cultural and spiritual connections to country. We recognize their valuable contribution to Australian and global society. Their contribution, especially to our understanding of native plants, and has been enormous. So, and goes over a very long period of time, about 70,000 years, which is a lot longer than colonization, which is just over 200. Now, there are a few housekeeping rules. So the presentation is to run for about 45 minutes. So in the seminar is from 12 to 1. After the presentation, we will hold a question and answer session. Please, if you have any questions, put it in the Q&A tab and not in the chat function. If you put them in the Q&A tab, I will read them out at the end, and Alistair can try to answer them then. Now, a little bit about Elliston Mctaggart. Alistair is a research fellow in the Centre for Horticultural Science. He studies the evolution of fungal pathogens. He focuses on rust and smut fungi in the past, and he has completed postdocs in Africa, America, and in Europe. So he's been everywhere. Alistair has started recently a new research direction at UQ, studying the biodiversity of psychoactive mushrooms in Australia. Normally, we would call that magic mushrooms. And his research will determine whether or not these Australian magic mushrooms have evolved here and have unique pathways for the production of psilocybin and confirm if they're endemic in Australia. Now, Alistair volunteered to give this presentation and came forward with a very punchy and short title, Sex and Drugs. Now, hopefully, Alistair will enlighten us whether or not there is a relationship between these two words, but to me it is obvious which one of the two he is most excited about. Alistair, please start.
I'm excited about them both. Let's be honest. Hey everyone, Alistair here. And I'd also just like to recognize Aboriginal Australians and the knowledge that they have about the use of mushrooms and fungi in general for foods, medicines, and biomaterials. And this talk seems like a perfect place to plant a seed or a question with you all, and that is whether cultures beyond Mesoamerica had knowledge of the use of psilocybin. Today, you'll learn about some of our native species of magic mushroom, or mushrooms that produce psilocybin, that have definitely been present here before humans arrived. Now, whether Aboriginal people knew about psilocybin will have impacts later under the Biodiscovery Act and possibly also under the Nagoya Protocol. So it's really, I think it's such a cool question. I'm not an ethnobotanist, so it's not something that I can answer or have the expertise to answer, but I think we should be discussing it. So yeah, let's kick on with it. If you don't know what psilocybin is, then it's possible that you've been comatose for about a decade, and welcome to 2022, where psilocybin is pretty much a monthly kind of thing that you'll see in the newspaper, if not weekly, with breakthrough potential in mental health. And I can't keep my finger on the pulse of every clinical trial because there are just too many. One of the ones that really stuck with me in the last 2 or 3 months is that in a trial to test the effect of psilocybin on people with depression, a third of the trial participants who took psilocybin were in remission for depression 6 months later after 1 dose, which is absolutely incredible. So psilocybin has this breakthrough potential for mental health conditions like depression, anxiety, addiction, PTSD. Palliative care is another area, and we haven't even explored the potential of psilocybin outside of human beings. In all of those clinical trials, the psilocybin used is synthetically produced, so it's not obtained from a mushroom, which is kind of an interesting thing in the industry. The value of psilocybin is $7,000 to $10,000 per gram, so it's a pretty lucrative space right now. And I'm aware of two, but I'm aware of a lot of people who want to join the industry, but I'm aware of 2 people who have permission to start producing psilocybin in Australia. I should say 2 companies. One is here in Queensland, our good friend Chris, and then there's another company in Western Australia who have permission to start producing psilocybin. And so it's hard to work with psilocybin because you need a permit to work on a Schedule 9 substance. So psilocybin is classified the same as heroin, which makes it an issue to work with. In fact, it's kind of the handbrake that stopped any research on psilocybin when I guess this scheduling came into play. Now, I got this permit. I think it's a little bit unusual because I'm not working on the compound itself. I'm working on the mushrooms that produce psilocybin. And the reality is that I don't have any psilocybin ever in the lab because I work on cultures of these magic mushrooms, so mushrooms that produce psilocybin. So most of the compound is produced in the pileus or the cap of the mushroom, and then a little bit in the stipe, and then very, very trace amounts in the mycelium. And I work with mycelium that only has that only has 1 copy of every chromosome. We're about to learn about that in a second. And mushrooms or fungi that only have 1 copy of every chromosome, if you're a Basidiomycota, then you're looking to have sex before you can start to produce those drugs. One thing about this permit, just to leave like a photo of a permit up on the screen, but it's pretty relevant and it's kind of cool because I think it's the first one in Aussie land to work on this. So Queensland, one thing that I've learned, Queensland has a really good system for penalising people who are in possession of psilocybin, quite a just penalty system where the Queensland Police Service will determine how much psilocybin you have, so the amount of psilocybin, whereas in New South Wales, they'll kind of extrapolate how much psilocybin you have from the weight of whatever they say has psilocybin in it. So if I was working in New South Wales and I didn't have this permit and all my cultures that don't even contain psilocybin were kind of weighed by the police there, they might say, oh, Alistair, you're in big trouble. Whereas in Queensland, which I think has a much better legislation and laws around psilocybin, will say, Alistair, you don't have any psilocybin, keep smiling. So that Schedule 9 compound as well, in the last 6 months, or maybe even less, My Medicine Australia and a group of psychologists and clinicians who are really passionate and driving therapeutic use of psilocybin they put in a proposal to downgrade psilocybin to a Schedule 8, which was an unsuccessful proposal, but those people who are passionate about the benefits of psilocybin for mental health, I think will persist, and it will be really, really interesting to see if it ever gets downgraded to Schedule 8, like cannabis was downgraded, and then that really kickstarted the industry. But I don't have a crystal ball, so I've got no idea what's going to happen. So you know that you can go to prison for having psilocybin in your possession. You also know that the clinical value is $7,000 to $10,000 a gram. The street value is pretty much, it's valueless. Like it's free. There's magic mushrooms are popping up all over Southeast Queensland. And like Andre and I were just talking about before we started, this rainfall is pretty much a great indication that there's about to be some mushrooms growing around town. In colder climates in particular, you'll have a different species of magic mushroom than up here in Queensland, where we have a prolific amount of Psilocybe cubensis, so these bottom two photos. Now, so you know the value, you know you can go to prison, but there's one more caveat, and that is this little group of mushrooms in this cluster up here, and that's a highly toxic group. And we're going to get to that later. So they look like psilocybe in that they are little brown mushrooms. So they can be quite confusing. So psilocybe is a genus of magic mushroom. So mushrooms that produce psilocybin. I'm just going to say magic mushroom for that term. There are other genera, about 4 or 5 other genera that produce psilocybin, but I just work on psilocybe, which is a monophyletic group. And every single species in that group produces psilocybin except for one taxon. So one species doesn't produce psilocybin. So we can kind of hypothesize that the most recent common ancestor of psilocybe, that most common ancestor could produce psilocybin. And psilocybin is just made, it's a product, it's a metabolic product when tryptophan, an amino acid, is metabolized into a tryptamine that then combine to serotonin receptors in mammals. And it's a cluster of 4 to 5 genes that perform this metabolic pathway. So that pathway was present in the most recent common ancestor of psilocybe. Now, in the most recent common, when we think about humans, we've got a sister genus, and that's chimpanzees. So psilocybe, they have a sister genus, and it's this little photo up here. That's Galerina. So a really closely related genus. Probably within the last 25 million years and above, they shared a most recent common ancestor. Now, Galerina don't have the pathway to produce psilocybin. They have a pathway to produce a different secondary metabolite, amatoxins. And amatoxins are present in Amanita, and it's death cap mushrooms. And that's the toxin that kind of causes organ failure and liver degradation and like a rapid and unpleasant death. So one really does not want to be ingesting mushrooms unless they know what they're picking and eating. One of the ways that you can differentiate Galerina, so this sort of look-alike and close relative of psilocybe, psilocybe, because of that psilocybin pathway, will stain blue when touched, and that's thought to be the oxidation of psilocybin into psilocin. So if you look on this stipe of this mushroom, you can see a faint tinge of blue. So you can handle a Galerina as much as you want, and it will never turn blue. If you take the top, the pileus off the Galerina, the cap, and you put that onto a piece of white paper, leave it for about 2 hours, it will drop its spores, and they will be a rusty brown color. Whereas everything in psilocybe produces a purpley brown spore print. And this photo in the bottom right is to illustrate that. So this big fellow right here is showering the guy underneath him with these purple brown spores. So you've got 2 characters that is really indicative of psilocybe, a purple spore print and a blue stain on handling. Now, just because you have a blue staining mushroom doesn't mean you have a hallucinogenic mushroom. Mushrooms that produce pores instead of gills, you would best wash your hands after handling one of those that had turned blue. Now, you'll notice in this top right image, this little shade of blue right here, and that is psilocybin being oxidized into psilocin, but it's not a species of psilocybe. That is Paniolus cyanescens, and it would be colloquially known in the community as a blue meanie. And it is not closely related to psilocybe. However, it shares an ecological niche. So all of these fungi pictured right now are saprotrophs. They degrade organic material. They're the reason that we're not up to our necks in wood all around. So they just degrade organic matter. They can live very easily on anything. You'll have other mushrooms that will form symbioses or mutualisms with trees, like mycorrhizae. They can't be grown easily in culture, but whereas saprotrophs, they can be grown in culture just on anything. So Paniolus, when you look at the group as a whole, that big group of Paniolus, only about 5 of them have the 4 to 5 genes required to produce psilocybin. And it's thought that in that shared ecological niche, in that big pool of microbial network, mycelium, that there's been an exchange of DNA at some point in time that's shared that pathway horizontally. So Paniolus or blue meanies acquired the genetic pathway to produce psilocybin horizontally. It's not the result of millions of years of evolution and then sharing vertically. They've acquired it from being in the same niche, most likely as psilocybe. Okay, so I think that's everything that I've just written there. The only thing I haven't mentioned is that Galerina and psilocybe are in the Hymenogasteraceae, which will definitely come in handy at your next trivia night. Since I started working on this, it's been Psilocybin cubensis has been the most prolific mushroom around. So its kind of season starts in late September, early October, and you can still probably collect it after the rain now. And it's pretty easy to identify by this annulus. So this is like a little strip of mushroom flesh that was once connected to the pileus, and as it opens up, it then gets showered and left there as a little skirt. And you can see in this one, there's a little bit of blue. So sorry, this is like a slideshow of where I've been collecting and whatnot over the last couple of months. When you get a cap back to the lab and you put it on a piece of paper or aluminium foil, this is that purple spore print that is really readily identifiable as purple. I'm sorry, I haven't made a spore print of Galerina to illustrate the difference of that rusty brown spore print. For those of you who are really savvy with color, you might have picked up that this spore print and this spore print are different colors. They're black. That's the spore print color of a Paniolus. And so when I was studying mycology in the United States, which is, I would kind of say is a bit further ahead of knowing what mushrooms they can and cannot eat in the Northern Hemisphere, they would say that you could pretty much eat a purple spored or a black spored mushroom. However, in Australia, we have such poor knowledge about what we can and cannot eat. You just don't want to be the exception to any sort of biological rule. When you put those spores under a light microscope, they'll look a little bit like this. Well, in fact, they'll look exactly like this. This spore here is 15 microns. I'm sorry, I didn't put a scale bar on there. Okay, the last little piece of information that you need before we get into it is a bit of knowledge about how mushrooms live, about their life cycle. So I've worked on Basidiomycota my whole career. And within Basidiomycota, which is a phylum of fungi, there are subphyla, the rust fungi, the smut fungi, and the mushrooms, Agaricomycotina. So I've worked on all of them. And what they have in common is that they spend most of their life cycle, in each cell, there'll be 2 nuclei, one from each of their parents. So one nucleus contains 1 copy of each chromosome. The other nucleus contains another copy of each chromosome. So you essentially have a diploid, but the nuclei are separated. So it's called a dikaryon. And in mushrooms, as you have that mitotic growth of hyphae or mycelium, those nuclei maintain fidelity by little clamp connections. So you'll have each nucleus will mitotically divide, and then one of them will go with the developing hyphae, and then the other will go whoop through a clamp connection to stay with its bud. we don't know how that occurs in rust fungi, because rust fungi don't produce clamp connections, but they still maintain their nuclear fidelity in a dikaryon. When you have a mass of mycelium or hyphae and you have an environmental trigger or the right environmental conditions, such as some delightful rainfall and you're in a piece of cow poo, that might be good conditions for Psilocybe cubensis, or it might be a lot of rainfall followed by a temperature drop. For the next mushroom we'll learn about, Psilocybe subaeruginosa, then the mushroom or the fungus will say, hey, it's time to form a mushroom, which really is saying it's time for sex, because this is the structure where sexual reproduction occurs. So in each of these gills, you've got millions of these little basidia. And so the basidium is the site of karyogamy, which is where these two dikaryotic nuclei fuse to form a diploid nucleus. And then when you've got a diploid, you can have meiosis. And each of these basidiospores gets a recombinant copy from the two parental nuclei. So that's kind of the pattern of sexual reproduction. Then, you know, the aim of everything is to reproduce. So you'll have two basidiospores and they're just trying to meet each other. They'll meet up, whether they're compatible or not. We're going to actually dig into sexual reproduction, trust me. Sometimes they'll form a new dikaryon and kind of realize, well, this isn't meant to be, and then they'll go their separate ways. We're not really sure what happens with psilocybe. So that's a normal life cycle. But look at this poor lonely guy over here. So this is what I work on. So I take basidiospores and then never let them meet each other. So I just want them as a true haploid. So only one copy of each chromosome. And that just makes genetics so much easier. And in Basidiomycota, haploid stages, which is just that necessary stage where they're trying to meet another haploid and have plasmogamy, which is kind of sexual reproduction in a way. They're just, everything is building up to that. So in smuts, you'll have haploid growth will be a budding yeast, and smut fungi are biotrophic pathogens. But as the haploid stage, they can't infect anything. They need to be together to get into a plant. With rusts, a haploid infection is pretty nondescript. It's barely even worth mentioning. It's the dikaryon that does the damage. Then with mushrooms, you'll have kind of flimsier growth. Maybe mushrooms that are pathogens won't be pathogenic, or they'll be weakly pathogenic. It's really a subdued life cycle. And I don't know if a haploid mycelium produces psilocybin or not. Mycelium have a very low concentration of psilocybin. It's possible that all of their attention is devoted to finding their sexual partner, but we don't know for sure. Okay, so my project, or our project, is on two mushrooms in Australia that are just on the East Coast that I can collect quite easily. This mushroom on the left, Psilocybe subaeruginosa, or in the a colloquial term would be to call it a sub, as in SUB, or a flying saucer mushroom, just for this kind of shape up here. Oops, sorry. I love this photo because it was sent to me by someone from Tasmania in her backyard, and I just love the purple spore print that you can see right here. So Psilocybe subaeruginosa was described in 1927 by a guy in South Australia, John Burton Cleland, JB. He described it from national parks in New South Wales, Victoria, and South Australia. And just keep in mind that this is in 1927. The pathway, or psilocybin as a compound, was known in Mesoamerica. We're 100% certain of that for centuries beforehand, but it wasn't in kind of the scientific literature until 1958, when it was described by Albert Hoffman. So JB Cleland didn't know that these were magic mushrooms when he was describing them. The other thing to think about is that in 1927, before, when I was saying we are behind compared to the United States of what we can and cannot eat here, we've actually applied a lot of names from the Northern Hemisphere to our Australian taxa, to our Australian mushrooms. So just keep that in mind. This mushroom, a very, very close looking thing to it, so something morphologically very identical, was then found in Kew Botanic Gardens. Oh, wait a second. Let me add one more thing. J.B. Cleland described this from wood chips, of eucalyptus wood chips and soil and grasslands. It also grows, I've collected it in rainforests in southeast Queensland. So a lookalike mushroom was described about 20 years later from Kew Botanic Gardens, which is essentially a multicultural melting pot for trees collected from all over the world and soil dragged from all over the world. And so this mushroom was described as Psilocybe cyanescens, cyan meaning blue. So it turned blue whenever it was touched. So alarm bells might be going off for you already. An Australian mushroom described in 1927, and then a lookalike described in the Northern Hemisphere, which is normally well ahead of Australian mycology. That mushroom in the Northern Hemisphere spread all throughout Europe, and it spread all up and down the west coast of the United States. It became one of the go-to species of magic mushroom for recreational use, and it became one of the taxa that catalyzed research on psilocybin. In fact, you can read about Psilocybe cyanescens patented in the 2000s for extraction of psilocybin and for treatment with psilocybin. So it's been patented, not Psilocybe cyanescens has been patented, not subaruginosa. So I am working on that particular mushroom. This mushroom over here, Psilocybe cubensis, or the gold top, is most likely an introduction to Australia. So the clue is in the name here. Ensis on the end of a Latin epithet just means from. This was described originally from Cuba. And I believe that it's kind of got this worldwide distribution now wherever cows are growing in subtropical and tropical environments. So one of my favorite pieces of literature, a guy who was visiting UQ from Scotland, Roy Watling, he talks about all over Indooroopilly, all over Long Pocket, which used to be a big cow farm, was covered in Psilocybe cubensis. He talks about it just being prolific all up in Caboolture and all over the Gold Coast. And he talks about how farmers would liven up a meal by adding a Psilocybe cubensis to whatever they were cooking up at the time. So it's possible that this taxon is native to Australia. Maybe it was in a niche of diprotodon dung or one of our big megafauna marsupials, just kind of living the dream out of that substrate. And then when cows were introduced here, it made a comeback. But because of its worldwide distribution, it's really hard to know where it came from. This mushroom, along with Psilocybe subaruginosa, is another go-to taxon for extraction and production of psilocybin. And in the recreational kind of world, this mushroom is spores are shared internationally. And it's usually what people are growing if they're illegally growing psilocybe. So it's unknown where its center of origin is. It's likely introduced to Australia. So I just spoke a lot, just with a couple of photos. I'm just going to put the key points up. So subs, or flying saucer mushrooms, were described from Australia, from natural environments in 1927. Then almost 20 years later, a really almost identical looking mushroom was described from Kew Botanic Gardens. And in the United States, it doesn't occur in native environments. In Europe, it doesn't occur in native environments. It's similar to Psilocybe cubensis here, which occurs in cow farms. It occurs in planted garden beds. In fact, subaruginosa you can find in a lot of garden beds in a temperature drop below the Queensland border. So both of them described before 1958. Yep, and I think you can remember that from Roy Watling. And these two taxa are the go-to for psilocybin. So this brings me to my research questions and kind of what I'm looking at. So with that permit, I've also become the first kind of safeguard of the genetic diversity of psilocybe in Australia. So everything I culture, I put into the herbarium as a living culture, which will last forever in glycerol. Then my question really is whether Psilocybe subaruginosa is native to Australia and introduced elsewhere into the Northern Hemisphere. And this is a population question as well, so I'll be answering it with genomics. So we want to look at subaruginosa and whether there is extra biodiversity than just one taxon. So other mycologists have come to Australia, not Australian mycologists, and they've divided Psilocybe subaeruginosa into 3 or 4 taxa, and then you'll have very close relatives in New Zealand as well, which we'll talk about in a little bit as well. So whether that should be all one thing, Psilocybe subaeruginosa, or whether it should be multiple species that maybe are structured by geography or substrate, whether they grow in a rainforest or a grassland, we're yet to find out. So, and then a third question is whether Psilocybe cubensis is exotic to Australia or not. And then if it is exotic, how many times has it been introduced? So that's really the crux of what I'm doing. Now, I just want to take you through a couple of the methods to get you familiar. This is a really uncommon method. If I notice a mushroom has stained blue heavily, which anecdotally means that it could have a high concentration of psilocybin, I will culture it directly from stems. So I'll do a surface sterilization, and then we expect the dikaryotic mycelium to grow out of that stipe. I don't really have many of these cultures. I have possibly 3 or 4, and that would be in there if anyone in the research community just needs a dikaryotic culture to fruit mushrooms from in the future, then they'll be able to go to those specimens. As I said before, what I really work on is the haploid, and that's because it's so much easier for genetics. So from the spores, I'll streak them out. This is an atrocious photo. I really should do better, but I'll streak them out so that it's just a biological technique, a 16 streak for those of you in the know. We're just moving spores kind of in a dilution to get them individually. And from that individual haploid spore, I can sub that and then have a haploid or mycelium with just 1 copy of every chromosome. So from there, we can grow it up in liquid for a very easy DNA extraction. So what am I sitting on? After the project's been going for less than a year, and maybe I'll get to work on it once or twice a fortnight. Luckily, I've been getting sent specimens as well. The project is not funded in any way, so it's all done creatively and through my own travel. Luckily, I love this topic, so, and I can, I've got money in bank accounts all over the world, so I can afford this kind of stuff. But yeah, I've got about 56 haploid cultures of Psilocybe cubensis from 7 populations, and for subaeruginosa, we've only got 2 populations, one thanks to Nigel Fechner, the mycologist at the Queensland Herbarium, who had collected one. Unfortunately, I couldn't make a spore print, so it's a true diploid cultured from the stem, and then I've collected from 1 population. And I've sequenced genomes just because I had a bit of spare cash lying around. And AGRF, thank you to Sarah, if you're watching, did some high throughput sequencing for me with Illumina reads. And now mushrooms, or fungi in general, the average genome size is probably 30 to 50 million base pairs. So if you're not in the genomics world, a human genome is 3.1 gig. So on a memory stick, it would take up exactly 3.1 gig. So these mushrooms are pretty tiny in comparison. So they'll take up 30 to 40 to 50 megabytes, which is the same as 30 million base pairs. And then the amount of data that you need, really, it can be done quite cheaply. So I'm sequencing these genomes for between 170 to $270 per genome, depending on how many I can do at any one time. So the genomes cubensis are these kind of puke green, I guess, and then subaeruginosa is this teal color. So we've got about a 10 million base pair difference between subs and cubes. The number in the middle is how many contigs I have from my assembly. As that number decreases, we're getting closer to the number of chromosomes. So you'll see just down the bottom, Psilocybe cubensis, a really nice long read assembly, has assembled about 13 chromosomes. So I'm well above the number of chromosomes. You'll see this one anomaly on the end here that's like 10 times, more than 10 times, got more than 10 times the amount of contigs and everything else. And that's Nigel's dikaryotic culture. So I did sequence 1 dikaryotic genome, and you can just see already that assembling it and dealing with it is kind of tricky compared to the haploids. So that really illustrates this tenfold difference between the number of contigs just means that the assembly is not as clean. So I will always be trying to work with haploids. Okay, so the next thing I just want to show you is that I got the right thing, that I sequenced the right thing. This is a ITS relationship, a way to visualize the relationship among different species of psilocybe. So I've just taken the ITS region. For those of you who don't know about the ITS region, essentially it's a molecular barcode. In all eukaryotes, we have ribosomes. We've got a big top of the ribosome and a bottom half, the small subunit and the large subunit. That's one long piece of RNA that comes out of the nucleus and binds with proteins to form a ribosome. In between the small and large subunit, you have a small piece of transcribed RNA that doesn't structurally form a part of the ribosome. You can imagine that because it's not responsible for translating messenger RNA into a protein, it doesn't matter if it starts to accumulate a couple of changes. So it's quite variable when you're looking at a species rank, although it's not perfect. But the fungal community, the mycological community, uses the ITS as the go-to region as a barcode to say, hey, this is probably what you're dealing with, or this is at least the genus rank. For us, we're probably getting down to species rank with psilocybe, and you'll see, so our teal green color here, this is a phylogenetic tree which enforces a bifurcating kind of phylogenetic hypothesis, evolutionary hypothesis. And these networks, these kind of satellite looking things, each circle is indicative of how many sequences have that same sequence without any parsimony informative changes. So 30 sequences in this Psilocybe cyanescens clade have the same sequence essentially. And in that Psilocybe cyanescens, or subaruginosa, I should say, that's a slip of the tongue. In this subaruginosa clade, you've got these 1, 2, 3, 4, 5, 6 different species. Whether they're true species or whether they're all subaruginosa is our question. And this is kind of an indication that we do have a little bit of work to do to tease out whether these are population differences or true species. So the two New Zealand taxa, Weroroa and Macarorae, they're also lookalikes to subs in New Zealand. It will be super interesting to see what is happening there as well. This little off-green thing is the Psilocybe cubensis group. This guy is in it. That's Psilocybe natalensis, which is described from South Africa or Africa. Now, the center of origin of Psilocybe cubensis, like I said, is unknown. And I think really as this starts to get more sampling from natural collections, it's going to be really interesting to see what happens. So most of the sequences in here are from the community who illegally grow magic mushrooms, or maybe not even illegally, because it's not illegal to grow them in Canada and Colorado. So it could be people populating data from there as well. And this is another way to visualize it, but I think it's a better way because we're not constraining it to a dichotomous hypothesis. So in this kind of a network, you're visualizing every single relationship among the data. And really the drive home point is that we've got this massive group of things that have different names, but whether we call them one thing or multiple things, that's a population question. And sort of the same for Psilocybe cubensis up here, although pretty clear that this is probably just one species with Psilocybe natalensis in this little triangle here. So networks are the way to go. I much prefer them over phylogenetic trees. Now, in summer semester, I had the pleasure of having a guest lecture in Liz Aitken's fungal biology course. Shout out to that course. Liz offers it to everyone in UQ, so you can sign up, I think, even if you're not a student. And I was asked a question, why does Psilocybe cubensis only occur on cow farms? And with confidence that I wasn't entitled, I said, it occurs everywhere. It's just that's the niche that it grows out of. And it wants that carbon and nitrogen rich substrate and a bit of rain to fruit. And if you put a cow poo on your backyard, maybe you'll have a Psilocybe cubensis grow. And I thought, after I finished the lecture, I wonder if I should check that. And I did. So using the Biomes of Australia soil environment sequences, which congratulations to the people from UQ who developed this, Paul and Phil and colleagues, you can virtually look anywhere in Australia at what microbes, bacteria and fungi are living in soil environments all across the country, and usually sampled from natural undisturbed regions. So super cool. Now, I expected when I looked at these data that Psilocybe cubensis was going to be everywhere, because I've collected it up in the Daintree, and I know it occurs as far south, almost as far south as Sydney. So it's pretty gregarious up and down the east coast of Australia. Now, on this phylogenetic tree, which is similar to the last ones, the same colors corresponding to our different taxa, so Subaruginosa in the teal, this green is cubensis. The bold is what you need to pay attention to. So the bold labels are base sequence sampling. And what you'll see is that from all those biomes in Australia, from over 3,000 collecting sites, there was not a single Psilocybe cubensis sequence among those data. We had Psilocybe subaruginosa. We even had it all the way over in Western Australia. We didn't, the base data didn't pick it up in Queensland, possibly because of where they sampled. They didn't get it in New South Wales, South Australia, or Victoria, which is a little bit unusual. And maybe that does speak to the sampling in a way. But yeah, so we've got these 1, 2, 3, 4 bold clades of fungi that occur in Australia in natural environments. And I would hypothesize that those are probably our native taxa. And what's super interesting to me is this one here, which hasn't ever been sequenced before other than in the base data. And it was collected from Darwin in Litchfield and Kakadu National Parks. So we only know it from sequencing data. We don't have a physical specimen. But what's really interesting is that one of the godfathers of Psilocybe, Gaston Guzman, who really was obsessed. Obsessed is the wrong word. He was an aficionado. He advanced the field so far. He had found this mushroom, Psilocybe brunniocistidiata, in Papua New Guinea. He'd actually described 6 or 7 mushrooms from PNG. And he found it on Nothofagus wood in a subtropical forest. And he hypothesized that this mushroom would be sister to Psilocybe youngensis. And I found it very, very interesting that this mushroom, that we have no idea what it looks like, but the sequences are sister to Psilocybe youngensis, and it occurs in northern parts of Australia. I found it very interesting that it had that sister relationship that Guzman predicted for this taxon, Psilocybe brunniocistidiata. So I would imagine, though, you know, it's all hypothetical until we physically have a spore print and a mushroom, I would imagine that it might look something like this if you're up in Darwin or anywhere in Northern Australia and you come across some sort of Nothofagus wood or any sort of decaying wood after a little bit of rain, then keep your eye out for this kind of a mushroom. And you now have the knowledge to identify Psilocybe as well. Okay, and now what we've all been waiting for, the sex. So one of the things that will come down to in industry, if I've got, say, diploid cultures that I can give to medical researchers who want to trial different strains of Psilocybe, that's fine. But because most of my collection is haploid, we need to cross it to be able to produce a mushroom and actually produce psilocybin. And so what you see here are 2 haploids side by side, and this is sexual reproduction. So this is that haploid changing into that robust mycelial growth that we all love so much. Whereas over here, those flimsy haploids were just never meant to be. So they do not get to be a dikaryon. Now, the question is, why? What's controlling this? And can we predict it from the genomes when we end up with a massive genomic resource that we can link to our massive culture collection? Oh, and this is going to be soporific. I recently spoke about this last year with my Rust talk. So you can tune out if you remember what's going on. But in Basidiomycota, you essentially have genes controlling mate recognition and mate compatibility. And these are homeodomain genes at a homeodomain locus, and then pheromone receptor genes at pheromone receptor loci. Now, whether those genes have to differ at every gene or not is one indication of whether, sorry, hold on, I should go back a notch. Those genes usually have to differ for mate compatibility. Similar to an X and Y chromosome in humans, for a compatible cross, you want an X and a Y chromosome that differ. So you could think of that similar to mushrooms. But with a lot of mushrooms and a lot of Basidiomycota, you almost have another two genders, because this pheromone receptor locus is sometimes also controlling mate compatibility. So you also, not only do you have to differ at that first locus, but you might have to differ at the next pheromone receptor locus. And in that case, you have tetrapolar mating. So when you've got a lot of genders, it's tetrapolar mating. When you only have to differ at one locus, you've got bipolar mating, similar to humans. Then there's all these different ways that this can be structured. So your pheromone receptor locus might be really close on the same chromosome as your homeodomain locus, in which case those genes are most likely linked, and that would be bipolar. In this image of Mycosarcoma matus or corn smut, our pheromone receptor genes occur on a different chromosome to our homeodomain genes and are not linked, and then they must differ in both the pheromone receptor genes and the homeodomain genes for a compatible cross. So a good characteristic of tetrapolar mating. Now, it's suspected that a lot of mushrooms had a tetrapolar ancestor. And one of the benefits of having tetrapolar mating is that you limit inbreeding. So, I feel like going back to those basidiospores, but essentially, you've got four sister basidiospores, and each will get one copy of a homeodomain gene. Sorry, two will get this copy and two will get this copy. So it will segregate. And then you'll have segregation of your pheromone receptor loci as well. And ultimately, tetrapolar mating is making it so that you can only cross with one of your sister basidiospores. In bipolar mating, you can cross with two. So that's the ultimate outcome of tetrapolar mating, is to limit inbreeding. So I just pulled out these loci from our genomes of Psilocybe cubensis and our population of genomes from Psilocybe subaeruginosa, and just wanted to see what was happening. And what was really interesting is that we have these three pheromone receptor loci in Arabic numerals in Psilocybe subaeruginosa, and we've got four pheromone receptor loci in Roman numerals for Psilocybe cubensis. Now, I think that my cubensis probably sampling needs to be increased, but we can see here that we've got segregation in a biallelic, at this biallelic gene, or this locus of this pheromone receptor locus, where half of our population has gotten one copy of the gene, and this and the other copy of the gene has gone into the other half. And then the same thing has actually happened up here. And now the chances are, and then this one, it's kind of hard to say what's happening in subaeruginosa down there. So this is a little bit confusing for me, because it looks like we've got segregation of two pheromone receptor loci, when normally you should really only have one segregating if it was true tetrapolar mating. And these loci are not linked in any way. I'm confident about that just because of the size of the assembly. So I need to talk to someone who knows more than me. The homeodomain locus is even more confusing to me, and that's because in our population from subaeruginosa, we have identical copies. We didn't really get segregation going on in that population. Number 208 is also from that population, and it got a different copy of the homeodomain locus, but it hasn't divided up cleanly in the population, which is what you would expect. Now, whether this is because I need to increase the sampling or whether something funky is going on, I'm not really too sure. I think if nobody else volunteers to keep talking, I will always volunteer to talk about psilocybe. And you'll just see up here, here's Galerina marginata. That's that sister to psilocybe that produces amatoxins instead of psilocybin. Okay, a big thank you to everyone who, since I've started the project, has collected me specimens or sent me spore prints or allowed me to be on their land. Big thanks to Miss Bunyip for sending me this. This is not a species of, this is a species of psilocybe, like it's super cool. I'm not sure what it is. Miss Bunyip identified it as Psilocybe cosamuiensis. I will put it into the herbarium, but it's still better that I receive spores, because whatever spores you send me, I will culture and put into a living collection, and they'll be there forever. The take-home messages from my talk. So, well, first, the first take-home message is that now you know how to identify psilocybe, just so that you can identify it and photograph it for me if you want to. The reason that I'm doing the project, I think, will have flow-on effects for biosecurity. Just as this psilocybin industry kicks on, they'll need to be importing new lines of Psilocybe cubensis or Psilocybe cyanescens, if that's the go-to taxon. I think it will stop biopiracy because a lot of our mushrooms are leaving the shore. So subs get sent internationally quite often. And it will help with the Biodiscovery Act. Eventually, one day, it would be great to link in this knowledge with the Biodiscovery Act in some way. So Psilocybe subaeruginosa, likely native to Australia based on its year of description, based on its habitat, so from natural areas in Australia compared to in the Northern Hemisphere where it's in introduced invasive areas. And whether there's more biodiversity in Psilocybe subaeruginosa will be a question answered by population genomics. Most likely, Psilocybe cubensis is introduced to Australia, and even just from the base data, it's super interesting to see that it hasn't occurred in any of the natural environments that they sampled. We had at least 2, probably 4, maybe even more hallucinogenic mushrooms that have been present in Australia for millennia, well before the arrival of humans. So that's something to chew on. That harks back to the question I asked you, or that seed I planted with you at the start of the talk. And likely the industry is going to have to work out how to cross these things, dealing with tetrapolar mating, if it's not even something even more than tetrapolar, based on those 2 segregating pheromone receptor loci. I want this research to be totally open, and it is. You can keep up with everything that I do on the blog. Like I said before, it's not funded in any way, shape or form. So you can try cracking the whip if you have a specific question that you want me to try and answer. But unfortunately, I have a real job. No, not unfortunately. I love my real job. Okay, thank you.
Well, thank you, Alistair, for a enlightening and entertaining talk. So please, if you have any questions, put them in the Q&A box. I may start off with one question. That is, if you look at plants, fungi, and bacteria, they do what I call chemical warfare to defend themselves from predation and from other organisms to eat them. So what is actually the role of psilocybin in nature? So what benefit does the mushroom get or derive from it, from having it? And what
I love that question, Andre. It keeps me awake at night. So the pathway for psilocybin evolved about 24 million years ago. That's a best guess based on molecular dating. I've just put up this image just because it's got this, what I photographed. This is up on the scenic rim, which I think was herbivory. So I was talking to a chemistry colleague about psilocybin being a deterrent, but I think we need to go back 24 million years ago to answer that question. To me, it's so hard to know. The fruiting bodies, so mushrooms are produced because they want to have sex, they want to be spread a lot of the time. So you either attract things, like maybe if you've got a stinky compost mound in the backyard and you see a stinkhorn develop, that's attracting flies to spread its spores. So mushrooms want to be picked, they want to be moved around to drop spores everywhere. Where the psilocybin evolved for mammals, I don't know, I can't answer. A lot of the time as well, you'll collect psilocybe and you'll see it swarming with little insects, all chewing on the inside. And it's always interesting to know what's going on in their little brains. One more little anecdote. I spoke to a farmer once. In fact, that time I was collecting in the Daintree, and he was a cow farmer, and there were magic mushrooms all over his property. And I said, do you know what these are? He said, no, he didn't. And I said, okay, well, they're magic mushrooms. And he asked me, do cows eat them? And I said, no. And then I got home and I thought, oh, wait a sec. Maybe cows do eat them, like accidentally when they're hoovering through long grass, they might get a hold of one. And he seemed convinced that his cows went loopy sometimes, which, I asked him if there was claviceps around. So claviceps, the cause of the precursor to produce lysergic acid, or LSD, is a pathogen of the inflorescences of many grasses. We have all sorts of native ones in Australia. And it causes cow staggers. So because it hasn't been purified into lysergic acid, it causes ergotamine poisoning and blindness and gangrene. He didn't have any of that around. Maybe it was magic mushrooms that his cows were going loopy from. I don't know.
Okay, Andrew, Alistair, you have opened Pandora's box because the questions keep rolling in. So you've got to give us some quick, short answers on some of these questions. Which came first, the Mesoamerican or the Australian psilocybin?
The center of origin of the mushrooms is believed to be in Mesoamerica. That's just because of the species diversity there. However, as we start to determine our biodiversity here, it may be that we find out that we've got a really biodiverse kind of group of magic mushrooms, especially like when you look at PNG, which has 7 or 8 species of magic mushroom, all psilocybe. It's kind of like, wow, this is a little hotspot of psilocybin mushrooms. So then the question is whether you can live with something for 70,000 plus years and not sample it when you're a culture of chemists who have worked out how to purify psychads and whatnot. So that's where that leads to.
Okay. So are there any benefits on harvesting psilocybin from mushrooms directly opposed to the chemical synthesized form like they do in the US?
Yeah, I think it could come down to cost. So mushrooms are pretty cheap to grow. They're one of my colleagues, Mickey, who works at Little Acre. His whole business is totally upscaling the byproducts of Australian wood and grain. So he takes all of that and he just grows it totally sustainably. And you could do the same with magic mushrooms. So you can make a completely ethical and sustainable industry just growing mushrooms. I think the challenges come down to really getting pure psilocybin and then storing pure psilocybin as mushrooms. I think that any industry based in Southeast Queensland will have a lot to contend with of just storing mushrooms to preserve that psilocybin content.
Yeah. What is the percentage dry weight psilocybin then in a mushroom?
It varies, and it's a phenotypic thing. I mean, obviously there's a genetic baseline, but so you'll have different quantities of psilocybin produced in different parts of the mushroom, and then some mushrooms will have 1% psilocybin per gram, and then others will be in the 0.9. So you'll still have a scale of psilocybin within normal phenotypic variation of an environmental population. So yeah, you would, if you were making an industry, you wouldn't do it from a clone. You would probably start with a lot of fast growers and high psilocybin content, and then really optimize the genetics to produce mushrooms that grew quickly, fruited prolifically, and had a high concentration of psilocybin.
Yeah. So someone has certainly an interest in this because he wants to know what the best areas in Brisbane are to go hunting for mushrooms after the rain.
In Brisbane? I wish I knew. I don't actually have any sampling from Brisbane. When I was a young man, I grew up in Sherwood. I could walk. I used to volunteer somewhere in Sherwood, and that was within 15 minutes of my house. And that was the first time that I ever saw a magic mushroom. So you could get them that close to Sherwood even. You need to go to a place for Psilocybe cubensis. Hold on, I don't think I should be talking about this. Psilocybe subaeruginosa, which is a wood chip lover. If you were living below Queensland, so I feel like a bit safer to talk about this. That occurs in mulched garden beds all down through New South Wales, Victoria, and Tasmania. So it's maybe head down there in the winter.
Okay, so the final question really is, why is Australia a few decades behind North America in mycological research?
There's probably like so many different reasons. One of them I would put down to critical mass. So I think right now where mushrooms are just such a hot topic, everyone is talking about them and how they're ethical and sustainable sources of food. And then with the magic mushroom industry as well, starting to gain so much traction, where I think we're starting to recruit more people. But when you go to a mycological congress in the United States or in Europe, you just see that there's, I mean, it's a bigger population, but there's just such a huge amount of people who are mushroom aficionados. And that's not to say that here, because we've just got people who are truly enthusiastic and passionate about mushrooms with so much knowledge. But, you know, Liz Aitken, we heard about her mycology course. I don't know how many mycology courses are offered, but that must be one of the few that's on offer. People are asking me where to study mycology all the time. I got into mycology through plant pathology, and then went to America and Europe and South Africa and picked up more as I worked with different people. So I didn't learn what I know in Australia. I don't know. It's a big issue. I wish we had critical mass.
Okay, thanks, Alistair. See, my personal experience with mushrooms is a little bit different, because I went to Germany once, and there was on the menu a dish called wild collected mushrooms. And when I ordered it, the guy said, "That's fine, you have to pay up front." Yeah. So,
yeah.
Anyway, thanks very much for your seminar, and very enlightening and very entertaining. So next week we have the next seminar is by Professor Damien Hine, and he's going to talk about narrow zero net emissions from the agricultural CRC bit. I hope you will listen to that. I know that Damien is an economist, but we won't hold that against him. It will be an interesting talk, because this is a big issue to go to net zero emissions by 2050. So thanks for attending the seminar today. If you haven't already signed up, you can, to our seminar invitation list, you can do so at our website. We would also like to invite anyone who watched our seminars to give us feedback. And you can give feedback by scanning the QR code, and that opens up a link. So thanks very much, and we'll see you next week. Bye.